Hey everyone, so today's topic for discussion is neoplasia. So neoplasia is very important topic for your NEET PG exams or other competitive exams as well as your professional exams, and many MCQs and long questions have been asked from this topic. So neoplasia, what does neoplasia means? The neoplasia, okay, this word is derived from two words. As you can see, one is the neo, another one is plasia. So what does neo means? Neo means anything new. Okay, new, a new is neo, and plasia does means growth. Now growth can be attributed to multiple things. Like the first one is increase in cell number. Okay. The second one is differentiation. Now what is differentiation? Differentiation is when a cell knows what is its function. Okay, so differentiation is cell will be now doing its function. Okay, differentiated for a particular function is differentiation. Now cell number increase, differentiation, and cell size. Okay, so increase in cell number, neoplasia, new growth. Okay, so cell number, here we are talking about new growth can be of cell number. Okay, mostly neoplasia does means a tumor, or tumor will grow when the cell number will increase. Okay, now what, when we will use plasia word for differentiation, have you heard of a term anaplasia? Okay, anaplasia does means, ana means no, plasia means differentiation. Okay, so anaplasia does means no differentiation. Okay, and another word, that is dysplasia. Dysplasia is abnormal growth. Okay, now abnormal growth can be of cell number, cell differentiation, or cell size. Okay, so this was a brief about what is actually neoplasia does mean. Neoplasia means a new growth, and most of the time this new growth is of cell number. Now this neoplasia can further be divided into two parts. Okay, the first one is benign and the second one is malignant. Okay guys, now what is the difference between benign and malignant? To understand this thing, let's discuss these two in a conceptual way. Okay, so suppose that this is one cell. Okay, this will divide and divide. Okay, now this is neoplasia. Okay, now suppose we have a border or a capsule that is surrounding these cells. Okay, now what will happen, this cell will continue to divide till it reach to the capsule. Now because of this capsule, this tumor will have a defined boundary. Okay, it will remain in this particular part, it will not migrate to any other part of the body. Okay, so first thing is this presence of capsule. Presence of capsule will limit the cell proliferation. Okay, limit cell proliferation. The second thing it will do is limit cell migration. So capsule will limit cell migration, and capsule, and the last one is capsule presence. Okay, so this capsule will attribute certain differences between benign and malignant tumors. What are they? The first one, benign. Benign will have capsule, malignant will not. Okay, now benign will be slow growing. Okay, there are many reasons for benign to be slow growing, but the uh, we will be remembering it here as because of capsule, benign will grow slowly, and there is no capsule in malignant, so it will grow fast. Okay, now the third one is the metastasis. That is very important property of a malignant tumor, that malignant tumor do metastasis. Okay, and benign does not. Okay, so, and anaplasia is absent in benign tumors and present in malignant tumors. So these are four main differences between a malignant and benign tumors. So we have talked about malignancy. And malignancy, this malignancy, the most feature of it is metastasis. So let's first discuss about the metastasis and how does metastasis do occur. Okay friends, so we will talk about metastasis, and invasion. Okay, or invasion. So let's first draw a malignant tumor, cells of malignant tumor. So this is some cells of malignant tumor that have no capsule or something that can stop them from metastasizing to another tissue. Okay, we will be talking here about hematogenous route of metastasizing. Okay, there are certain routes by which a malignant tumor tissue can metastasize, that we will discuss later in this video. Okay, so this is our blood vessel, and this here in between them is extracellular matrix. Okay, so this is ECM, this is blood vessel, and this is tumor tissue. Okay, coming down, what is difference between this tumor and this tumor? Okay, so this is tumor tissue. Okay, now these cells will detach from each other by decreased expression of E cadherins. Okay, so first point we have to remember is decreased expression of E cadherins. Okay, E cadherins. Now the second thing that it does, it gets attached to the extracellular matrix by integrins and combination with laminin and fibronectin. Okay, let's write it down. This is our extracellular matrix, so these cells will get attached here. Okay, the extracellular matrix via, what are they? Integrins in combination with, let's write it down, integrins in combination with laminin and fibronectin. Okay, phyla. Okay, phyla, fibronectin and laminin. Okay, so we have discussed two processes here. First, it is detached from the basic tumor cells. Here it is detached from here via decreased expression of E cadherin, then it will go and attach to extracellular matrix via these, what are they? They are integrins with combination of fibronectin and laminin. Okay, so now after this cells, this is extracellular matrix. Okay, and these are the tumor cells. So these cells will break down the ECM and basal membranes, will break down, will disrupt. Okay, will disrupt this ECM and basement membrane via two things. The first one is, So and this is point number 3. The first one is that are matrix metalloproteinases. Okay, matrix metalloproteinases. Metalloproteinases. Okay, so what are they? They are two types of, okay, they are of two types, 9 and 2. So matrix metalloprotein 9 and proteinases 9 and 2 will help these cells to disintegrate extracellular matrix as well as the basement membrane. Now this, from these two, the ninth one is more important than second one. Okay, now this one more enzyme is here that helps in this disruption, that is type 4 choline. It is type 4 collagenase enzyme. Okay, collagenase enzyme. So there are two enzymes that will help in this third process, which is disruption of extracellular matrix and basement membrane. So these are matrix metalloprotein 9 and 2, matrix metalloproteinases, okay, 9 and 2. Then most important one is 9, then 2. Okay, and then type 4 collagenase enzymes. Okay, so this will disrupt this ECM and basement membrane. So what will happen then, that after disruption of these extracellular matrix, these cells will have access towards the blood vessel. Okay, so it can easily go and penetrate in the blood vessel. Okay, so these cells will go inside the blood vessel. So here let's draw the diagram. So this cell goes inside the blood vessel. Here it will find some cells that we already know, that is platelets. So these tumor cells will combine with these platelets and these will form the tumor emboli. Okay, this will form the tumor emboli. Now what will this emboli do? It will further migrate towards two locations. Okay, it can migrate towards lymph node or can migrate towards any organ. Okay, so this was basic process by which a tissue metastasizes. Okay, let's again revise it. So metastasis or invasion, there are three things: tumor tissue, extracellular matrix or basement membrane, and blood vessels. Okay, so this tumor tissue will first disintegrate from its base, and this will happen via decreased expression of E-cadherins. Then it will attach to extracellular matrix or basement membrane via integrins, that is in combination with phyla. Phyla is fibronectin and laminin. Okay, after attaching with the extracellular matrix and basement membrane, this will disrupt these two via matrix metalloproteinases 9 and 2 and type 4 collagenase enzymes. Okay, after that it will go inside the blood vessel. It will attach with the platelets there and will form tumor emboli. Now this tumor emboli will further migrate to organ or lymph nodes. Okay, so this was the basic mechanism by which metastasis do occur. Okay, so benign and malignant, we will be further dividing it into epithelial and mesenchymal. So any benign tumor that have epithelial origin will be placed here. Any benign tumor that have mesenchymal origin will be placed here. Okay, so what are the examples of this benign epithelial tumors? The first one is papilloma. Okay, so papilloma and adenoma. Okay, now as you can see, there is one suffix that is present here, that is oma. The oma is particularly given to benign tumors. Okay, now the question arises, what are the malignant tumors with oma suffix, that we will be discussing, okay, later in this video. Okay, so papilloma and adenoma, these are two examples of epithelial benign tumors. Now, what are other examples of mesenchymal benign tumors? That is lipoma, okay, and osteoma, chondroma, fibroma. They all have oma. Okay. Now, as we have divided benign into epithelial and mesenchymal, we can also divide this malignant into benign and into epithelial and mesenchymal. So any malignant tumor that have epithelial origin will be placed here. Any malignant tumor that have mesenchymal origin will be placed here. Okay. So what is epithelial? Now, what are the suffix used for malignant tumors? They are carcinoma. Okay, they are carcinomas and sarcomas. So we can place epithelial. We can say SCC is an example of epithelial malignant tumor. Okay, APC squamous cell carcinoma. Okay, TCC can also be placed here. So as TCC is transitional cell carcinoma. Okay, transitional cell carcinoma. Now, if we talk about mesenchymal malignant carcinomas, mesenchymal malignant tumors, so mesenchymal will be sarcomas. Okay. Now, there are different types of sarcoma that we will be discussing when we will talk about systemic pathology. Okay, systemic pathology and tumors related to them. Now, malignant tumors that have oma suffix, that here are some examples of these exceptions that are like seminoma. Okay, the other one is lymphoma, chloroma, melanoma. Okay, now these are the examples of malignant tumors that have suffix oma. Okay, now okay guys, so let's talk about mixed tumor. Okay, what is a mixed tumor? So mixed tumor is derived from two or more than two types of tissues. Okay, if it is derived from two types of tissues, it is called as biphasic tumor. Okay, so biphasic tumor is a type of mixed tumor that is derived from two types of tissues. Okay, and now this mixed tumor can be derived from more than one germ layer. More than one germ layer or single germ layer. Okay, now examples of a mixed tumor that is derived from single germ layer is pleomorphic adenoma. And examples for a mixed tumor that is derived from more than one germ layer is teratoma. Okay, that is called teratoma. Now, teratoma can be of three types, that is cystic, mature, and immature. Okay, so guys, mixed tumor that is derived from more than one type of tissue. If it is derived from two type of tissue, it is called biphasic. It can be derived from more than one germ layer or single germ layer. If it is derived from single germ layer, what is the example? Pleomorphic adenoma. If derived from more than one germ layer, it is teratoma, and teratoma can further be divided into cystic, mature, and immature. Now, let's talk about some terms here. So now, what is choreostoma? Choreostoma means a normal tissue that is present in at abnormal location. Okay, no, tissue is normal but it is present at abnormal sites, like stomach. It is stomach. If pancreatic tissue is present inside stomach, it will be called as choreostoma. So choreostoma is normal tissue at abnormal location. Okay, now what is hamartoma? Hamartoma is disorganized tissue in normal location. Okay, location normal. Okay, and the tissue is disorganized. Okay, so now this disorganized tissue can be considered as tumor if it passes the hallmarks of a tumor. Okay, that we will be discussing later. So hamartomas are, so choreostoma and hamartoma. Choreostoma means normal tissue at abnormal sites, abnormal location, and hamartoma disorganized or abnormal tissue at normal location. Okay, so this was about choreostoma and hamartoma. Now we will be discussing about so we have previously discussed about anaplasia. Okay, what was anaplasia? Anaplasia means ana means no, plasia means a differentiation. Okay, so this is a hallmark of a malignancy. So there is a hallmark of malignancy, that is anaplasia. If we talk about the most characteristic feature of a malignancy, that will be metastasis. Okay, so hallmark of malignancy is anaplasia, that is lack of differentiation. Now other hallmarks of a malignancy can be, first one is anaplasia, second one pleomorphism, third one NC ratio high. So what is normal NC ratio? That is 1:4, and it will become 1:1 or 2. Okay, in malignancies. Okay, now hyperchromatic nuclei, that is also a characteristic feature of these cells, that is hyperchromatic nuclei. So what does it mean? That this cell nucleus will be more blue. Okay, more bluish in the color. Okay, hyperchromatic nuclei. The third one is prominent nuclei. Okay, and two more, loss of polarity and abnormal mitosis. So this can be asked in your theory exams, that what are the hallmarks of a malignant tumor. So that are anaplasia, pleomorphism, NC ratio high, hyperchromatic nuclei, prominent hyperchromatic nucleus, prominent nucleoli, and loss of polarity and abnormal mitosis. Okay, abnormal mitosis. So now we will be discussing about, before that let's discuss about some exceptions. Okay, there are two exceptions. So actually all malignancy does show metastasis, but there are two exceptions for this one also, and that are glioma and BCC, basal cell carcinoma. So these are two malignancies that does not show metastasis. Okay, so metastasis, these exceptions are always considered to be MCQ points. Okay, so always remember that malignancies are malignancies does have metastatic properties, but there are two exceptions for this, that are glioma and basal cell carcinoma. Okay, so now we will be discussing about further we will be discussing about metastasis. Okay, so we have already discussed about the mechanism of metastasis, so let's discuss some more thing about it, that is metastasis. So what are these things? So how does one tissue will metastasize to another tissues, okay, another locations? How does it will do it? Okay, so it can do it by certain mechanisms, certain pathways. Okay, so what are the pathways by which something travel in our body? So one is lymphatics, okay, the other one is blood vessels, okay, and body cavities can be one medium, and translomic can be uh, malignant tumor can also spread via translomic medium. So these are mostly four methods by which one malignant tissue will metastasize to other locations. So lymphatics and hematogenous route, then direct spreading via body cavities, translomic. One more method by which tumor can spread is via CSF. Okay, so we will be talking about these one by one. So the first one we will talk about is malignant tumors that spread via lymphatics. So what are the examples? Actually, most carcinomas do metastasize by this method, okay, via lymphatics. Okay, but there are always exceptions there. Okay, so except, what are the exceptions? One is SCC, hepatocellular carcinoma, RCC, renal cell carcinoma, and choriocarcinoma. These carcinomas do spread via hematogenous route. Okay, so most carcinomas will be spreading via lymphatics, but these three will be spreading via hematogenous route. Simultaneously, if we talk about hematogenous routes, most sarcomas will be spreading via hematogenous routes, but there are always exceptions. So what are the exceptions? Except alveolar rhabdomyosarcoma. Okay, alveolar rhabdomyosarcoma, and the one is synovial sarcoma. Okay guys, so there are two tumors that will be spread via lymphatic routes. Okay, and they are alveolar rhabdomyosarcoma and synovial sarcomas. All sarcomas will be spread via hematogenous route. Now direct spread via body cavities. The third one is direct spread via body cavities. I know I have a very poor writing. Okay, so what is the example for a malignant tumor that is spread via direct seeding in body cavities is pseudomyxoma peritonei. Okay, that is pseudomyxoma peritonei. Okay guys, now malignant tumor can also spread via translomic medium, but they are very less example for this, so we will not be wasting our time in this. Okay, translomic. Now comes the tumors that spread via CSF. Okay, this is a very important question that can be asked in your theory exam as well as your PG exams. Okay, so what are the tumors that spread via CSF. So we have two most tumors that spread via these via CSF, that is medulloblastoma and ependymoma. Okay, ependymoma. So there are two tumors that will be transmitted via CSF in body from one side to another side, that is medulloblastoma and ependymoma. Now there is one question that has been asked: what is the type of metastasis that is acquired by medulloblastoma to spread? Okay, so this is a drop. Okay, this is called drop metastasis to coda equina. Drop metastasis to coda equina. We will be talking about drop metastasis, okay, later in our part 2 video. Okay, coda equina. Okay guys, so drop metastasis to coda equina, there are two parts. There are two tumors that have been transferred from CSF via CSF. Okay, medulloblastoma, ependymoma. Medulloblastoma will show drop metastasis in coda equina. Till now it is clear. Now there are certain tumors that go spontaneous regression. Okay, what are the tumors that go under spontaneous regression? Tumors that undergo spontaneous regression. That are, again sorry for the writing. Okay, so that are MNC R square. Okay, MNC R square. Now what are the names? Malignant myeloma. It will be spread via, it will be showing spontaneous regression. Now neuroblastoma, choriocarcinoma. Now what is tumor that starts from R? Very famous RCC, renal cell carcinoma, and other one is retinoblastoma. Okay, these are very famous tumors. So retinoblastoma. One thing I want you to remember here, from retinoblastoma, remember RB. Okay, RB. We will be discussing the actual need of this RB, okay, later when we will talk about genetics of different types of tumors. Okay, so till now we have discussed many things like okay, how a tumor metastasizes, what are the root of metastasis, what are the tumors that go spontaneous regression. We have remembered it via MNC R square. Okay,